Imagine for a second that you're a detective.
Right.
You're trying to catch a criminal.
Okay, I'm with you.
But every time you get a description of the suspect, it changes. Like, one witness swears the suspect is a bone disease, brittle, painful, deforming.
Right.
And then another witness is absolutely convinced it's
A muscle disease.
exactly
Yeah.
a muscle disease. They saw the suspect causing weakness, trouble walking, trouble lifting things.
And then a third witness comes in and says, no, you're both wrong. This is a brain disease. It attacks the motor neurons. It looks exactly like ALS.
Right. And the crazy part is, all three witnesses are telling the truth. They're describing the exact same perpetrator.
Which is the terrifying reality of what we're discussing today. It's a medical shapeshifter.
Welcome to the deep dive. Today, we are wrestling with a beast of a topic known as multisystem proteinopathy, or MSP.
Oh.
And specifically, we're looking at a groundbreaking new study out of Huashan Hospital in China that fundamentally changes how we identify this shapeshifter.
It really does. This is one of those studies that forces doctors to rewrite the textbooks, or at least rewrite the chapter on diagnosis. Because for a long time, we thought we knew what MSP looked like. We thought we knew its classic disguise.
Right.
But this data suggests that in different parts of the world, the disease wears a completely different mask.
Before we get into that plot twist, and it is a big one, we need to ground ourselves in what MSP actually is. Because multisystem proteinopathy sounds like, well, a mouthful of jargon.
It is a mouthful, but the name actually tells you exactly what's going wrong. Multisystem means it doesn't just attack one part of the body. It can attack the muscles, the bones, the brain, the nerves. And proteinopathy means the root cause is a problem with protein. Specifically, the body loses the ability to recycle and clean up old proteins, so they just start clogging up the works.
We'll get into the mechanics of that clogging later because it's fascinating. But first, let's talk about the four faces of this disease. In the source material, they highlight four distinct conditions that MSP can mimic.
Right. Think of these as the four disguises. First, you have IBM, or inclusion body myopathy. That is pure muscle weakness. The muscles degenerate, usually in the limbs.
Okay, so phase one is muscle.
Phase two is the heavy hitter. ALS, or amyotrophic lateral sclerosis. This is a motor neuron disease. The nerve cells that control your muscles die off, leading to paralysis and, unfortunately, usually death.
So that's the brain and nerves. Phase three.
Phase three is FTD, or frontotemporal dementia. This isn't like Alzheimer's where you forget where you put your keys. This affects the front of the brain, so personality, behavior, language.
Wow. And finally, phase four.
PDB, or Paget disease of bone. This causes accelerated bone turnover, deformities, and pain.
So you have a single genetic disease that can look like muscle failure, nerve failure, dementia, or bone disease. That is pleiotropy in a nutshell, right?
Correct.
One gene, many outcomes.
Exactly. And that brings us to the study at hand. The researchers at Huashan Hospital did a retrospective analysis spanning 24 years from 2000 to 2024. They look at a massive cohort of nearly 1,000 patients, 953 to be specific.
And these weren't just random people. These were patients who walked into the neurology department with unexplained symptoms. They had ALS-like issues or muscle weakness or dementia, but no clear cause. The researchers were hunting for the MSP needle in that haystack.
And out of those 953 people, they found 29 confirmed cases of multisystem proteinopathy. That's a prevalence of about 3% in this specific high-risk group.
3% might sound small to some listeners, but if you are a doctor seeing 100 confusing cases a year, 3 of them are this genetic time bomb.
Precisely. And for those 3 people, getting the diagnosis right is everything. It stops the endless testing, the wrong treatments. It gives an answer.
Let's talk about who these patients are. What's the profile?
In this Chinese cohort, the disease leaned heavily male. About 72% were men. And the age of onset is really tough to read about. The median age was 44.
44?
Yes, mid-40s. These are people in the absolute prime of their lives. They're raising kids, they're at the peak of their careers, and suddenly they start tripping over their own feet. Or they can't button their shirt, or they start having trouble speaking.
Now, this is where the study gets revolutionary. You mentioned earlier that we have these Western or Japanese assumptions about the disease. What was the old assumption?
Well, if you looked at the literature coming out of Japan or Europe historically, the story was that MSP usually starts with the muscles. Inclusion body myopathy, that muscle weakness, was considered the dominant phenotype.
So if I'm a neurologist operating on that old data, I'm waiting to see a patient with weak muscles before I even think about MSP.
Exactly. You're looking for a muscle disease. But Huashan Hospital found something completely different. In this Chinese population, the dominant face of the disease wasn't the muscles. It was ALS.
That is a massive shift.
It's a complete flip. Out of the 29 patients they identified, 20 of them, nearly 70%, presented with ALS symptoms first. Only 10 had the classic muscle weakness onset.
So in China, MSP is masquerading as a fatal motor neuron disease much more often than it presents as a muscle condition.
Correct. And think about the implications of that. If a patient comes in with ALS symptoms, doctors usually test for standard ALS genes. They might not even think to test for MSP because, in their mind, MSP is a muscle thing. The study says, stop, you are missing the diagnosis.
So why the difference? Is the disease just behaving differently because of the environment, or is there something genetic going on?
It's the genetics. The underlying mutation profile in this Chinese cohort is distinct from what we typically see in the West.
Let's break down the big three genes they found. Who is the main culprit in this story?
In this study, the number one genetic driver was a gene called ANXE11 or annexin A11. It accounted for roughly 34.5% of the cases.
Annex 11. Okay. And how does that compare to the usual suspect?
The classic MSP gene is usually considered to be VCP, or volition-containing protein. In this study, VCP came in second at roughly 21%, and in third place was a gene called OPPN, or optineurin, at about 17%.
Were there any other players?
Yeah, they also found SQSTM1, MATR3, and HNRNPA1, but they were definitely less common.
So Annex 11 is the heavyweight champion in this specific group. And I'm assuming there's a connection here. Annex 11 is the reason we're seeing so much ALS.
You hit the nail on the head. This is the genotype-phenotype correlation. The study found that if you have the ANXA11 mutation, you are significantly more likely to present with ALS symptoms. If you have the VCP mutation, you're more likely to see the muscle weakness or bone issues.
So the gene dictates the disguise.
Exactly.
There's another data point here that I found really tricky. We call this multi-system proteinopathy. So logically, I assume the patient walks in with multiple systems failing at once. Bad bones, bad brain, bad muscles.
That is the logical assumption, but biology is rarely logical in that sense. The study found that over 2/3 of these patients only had one symptom type for a very long time.
So they didn't look like multi-system patients at all.
No, they looked like single-system patients. A man comes in with just ALS symptoms, or a woman comes in with just muscle weakness. Only 8 out of the 29 patients had that classic textbook mix of multiple systems failing simultaneously.
That makes the detective work incredibly hard. If I just have ALS, why would you test me for a syndrome that involves bone disease?
Exactly. It's the ultimate camouflage. The disease is hiding its true nature. And that is why the authors of this study are pushing so hard for broad genetic testing. You cannot rely on clinical observation alone because the symptoms are too isolated in the early stages.
And this is but the study did offer one really cool tool for the detectives out there. They found a pattern in the limbs that can actually predict the gene. I loved this part. It felt like Sherlock Holmes deduction.
This is arguably the most clinically useful finding in the paper. It gives doctors a physical clue to predict the genotype before the lab results even come back.
So let's role play this. A patient comes into your clinic. They're having trouble walking. Their legs are weak.
If the symptoms start in the lower limbs, the legs, it is a massive signal for VCP. In this study, 100% of the patients with VCP variants started with lower limb involvement.
100%. You never see 100% in medicine.
Almost never. It's a striking statistic. If the legs go first, you should immediately be thinking VCP.
Okay, now swap it. What if the patient walks in fine, but they can't lift their arms to brush their hair? Or maybe they're slurring their speech and choking on water.
That points directly to ANXA11 or OPTN. Patients with these variants predominantly started with upper limb involvement, the arms, or what we call bulbar symptoms, which is the speech and swallowing machinery.
That is fascinating. It's like the gene have a preferred target on the body map.
It does. VCP goes low, ANXA11 goes high.
And this isn't just trivia. This distinction matters for the patient's future, doesn't it? Because we established that ANXA11 is linked to ALS, and ALS is a much faster beast.
It is. The study looked at disease progression, specifically survival rates, and the need for mechanical ventilation life support. The difference was stark.
How stark?
In this specific snapshot of time, 60% of the ANX11 group had reached a hard endpoint, meaning they had either died or needed a ventilator to breathe.
And the VCP group?
0%.
Wow. 60% versus 0%.
No, we have to be careful. VCP is still a serious progressive disease. In this cohort, Annex 11 is driving a much more aggressive rapid decline because it is attacking the motor neurons directly.
So knowing which gene you have tells you how much time you might have. It tells the doctor how fast they need to move with interventions.
Exactly. It changes the urgency of care completely.
Let's zoom in a little further. We've talked about what the doctor sees, the legs, the arms, the symptoms. But I want to understand the proteinopathy part. What is actually happening inside the cells? Why do these different genes all break the system in the same way?
It all comes down to what we call protein quality control.
Okay, let's unpack that.
Imagine your cells are like a busy factory. This factory is constantly building proteins to do work. But like any factory, it produces waste. Old machinery breaks or a product comes out defective. You need a sanitation crew to come in, bag up that trash, and recycle it.
So the cell has a trash disposal system.
Right. In scientific terms, this involves the ubiquitin proteasome system and autophagy. Autophagy literally means self-eating the cell, eating its own waste to clean up.
So in MSP patients, the trash compactor is broken.
Exactly. The foreman of the sanitation crew is asleep on the job. The gene mutation, whether it's VCP or ANXA11, disrupts this disposal process. So what happens? The crash starts piling up.
And if you don't take the trash out, eventually the house becomes unlivable.
Precisely. These junk proteins form clumps or aggregates inside the cell. They physically clog up the cellular machinery until the cell becomes toxic and dies.
And we can actually see this trash under a microscope, right?
Yeah.
The study mentions rimmed vacuoles.
Yes. If you take a muscle biopsy from these patients, you often see these rimmed vacuoles. Imagine looking at a muscle fiber and seeing an empty space, a bubble, surrounded by a gritty rim of material. That material is the protein trash pile. It's the classic hallmark of inclusion body myopathy.
But because nature loves to throw curveballs, there was an exception in the study.
There is always an exception. They mentioned patient P20. This patient had the ANXA11 mutation, our new bad guy. But when they looked at his muscle biopsy, no rimmed vacuoles.
So if a pathologist was just following the textbook rule of no vacuoles means no MSP, they would have missed him.
They would have. Patient P20 had protein clumps, specifically P62 and TDP43, but he didn't have the classic visual sign. It's another warning to doctors. Do not rely on just one piece of evidence. The pathology can lie, or at least tell a partial truth.
Speaking of complex cases, I have to bring up the triple threat. The study details a specific patient, patient P2. This guy seemed to have everything go wrong at once.
Patient P2 is the poster child for the multisystem proteinopathy. He was a 53-year-old male. He had the muscle weakness, IBM. He had the bone deformities, Paget disease. But he also had something else.
Parkinson's disease.
Right.
Parkinson's wasn't on our list of the 4 faces earlier.
It wasn't because it's incredibly rare in MSP. It's usually only linked to VCP mutations, which this patient had. But just think about the burden on that patient. You have a movement disorder originating in the brain with Parkinson's, a degenerative muscle disease, and a metabolic bone disease, all caused by one spelling error in one gene.
It's terrifying, but it really drives home why they call it pleiotropic.
It highlights the diagnostic gap. If I'm a doctor treating his Parkinson's, I'm focused on his dopamine levels. I'm probably not checking his bone density or sequencing his muscle genes.
Which brings us to the big takeaway of this study. The authors are pretty clear about what needs to change in the medical community.
They are. They are arguing for the aggressive use of NGS next-generation sequencing.
Basically, stop guessing and read the DNA.
Right. Because the symptoms overlap so much, and because, as we learned, patients often only show one symptom for years, clinical observation isn't enough. You can't just look at a patient and know.
And specifically for the Chinese population, this study puts a big neon sign flashing over ANXA11.
Correct. If you are a clinician in East Asia or treating patients of Chinese descent and you see unexplained ALS or muscle weakness, ANXA11 needs to be at the top of your suspect list. You can't just test for the standard Western panel.
It effectively redraws the diagnostic map.
It does. It reminds us that diversity in research isn't just a buzzword, it's a medical necessity. If we only study genomes in Oxford or Boston, we are literally blind to how diseases present in Shanghai. We build a biased textbook.
And this study corrects that bias.
Mm-hmm.
It proves that ANXA11 is a major driver of ALS-like MSP in this region.
Exactly.
Before we wrap up, I want to leave you with a thought that came up when I was reading this. We talked about variable expressivity. That's the fancy term for how the same gene causes different problems in different people.
Right. Why does patient A get muscle weakness and live for decades, while patient B gets ALS and dies in 3 years when they have the exact same mutation?
It feels like a lottery. But it can't be just luck, can it?
Probably not. This is the next frontier of the research. Scientists believe there are hidden factors. We call them modifier genes.
Modifier genes?
Think of them as the supporting cast. The main mutation, ANX11, is the villain, but maybe you have a hero gene somewhere else in your DNA that protects your motor neurons. So even though you have the villain, the hero saves your nerves and you only get the muscle weakness.
Or maybe you have a traitor gene that makes your brain more vulnerable.
Exactly. And environmental factors likely play a role too. Exposure to toxins, lifestyle, viral history, these could be the triggers that decide which face the disease wears.
It's an unsettling thought that our fate isn't written in just one gene, but in the complex interaction of thousands.
Unsettling, but also hopeful.
How so?
Because if we can find those modifier genes, the ones that protect the nerves, maybe we can mimic them with drugs. Maybe we can find a way to force the disease to take the muscle weakness path instead of the ALS path.
Transforming a fatal disease into a chronic, manageable one.
That is the ultimate goal. And studies like this one from Washington Hospital are the first step. You have to identify the pattern before you can hack it.
Well, we've covered a lot of ground today. From the shape-shifting nature of MSP to the specific lethal role of ANXA11 in China.
We've seen how VCP targets the legs, how Annex A11 targets the arms and throat, and why we need to look beyond the microscope and into the DNA.
A huge thank you to the research team, Xia, Chen, Sun, and everyone at Washington Hospital for this rigorous work. It's dense science, but it translates directly to saving lives by getting the diagnosis right.
Absolutely.
And for you listening, thank you for diving deep with us. It's a reminder that even in the age of advanced medicine, our bodies still have secrets. But with research like this, we are decoding them one by one.
We certainly are.
We'll see you on the next deep dive. Take care.